Hey everyone, welcome back. We are in lesson 2 of week 1, and today we are covering translation. And before we start, we're going to start with our mindful minute. So I encourage you guys to sit with a mindful body wherever you are, and we'll take deep breaths together for 1 minute. You can either watch the animation, inhale and exhale, or you can close your eyes or look down if that's better for you. So, everyone together, inhale through your nose, exhale, inhale through your nose, exhale, inhale, fill your lungs, exhale, inhale, we're halfway there, and exhale, inhale, fill your lungs with oxygen, exhale, pushing out that carbon dioxide, inhale, and exhale. And last one, inhale deeply and exhale fully. And allow your breathing to return to normal. Check in with yourself. Has anything changed? Check in with your emotions. Check in with any physical sensations. Or maybe it stayed the same. When you're ready, you can slowly open your eyes. You had your eyes closed. All right, so the do now for today. 1, transcribe the DNA sequence into mRNA. 2, where does transcription occur? And 3, why is transcription important? So you guys can pause the video right now and try questions 1, 2, and 3. Again, you should have your notebook or piece of paper, and whenever you see this symbol in the top right, that means you are writing. So to transcribe our DNA sequence into mRNA, we use our base pairing rule. And for mRNA, if you remember from lesson 1, whenever there's an A, mRNA will pair with U for uracil because there's no T for mRNA. So we have U, T pairs with A, G pairs with C, C, C pairs with G, T pairs with A, A pairs with U, C with G, we got U again, and C and A. All right, where does transcription occur? Transcription occurs or happens in the nucleus. And 3, why is transcription important? Transcription is important because it's the first step of protein synthesis. Our learning target for today is I can transcribe and translate a protein from a DNA sequence. Pause the video now if you need to write down the learning target. So in lesson 1, we looked at 2 things that DNA could do. DNA can replicate or make a copy of itself, and it can also be transcribed into RNA. So DNA serves as the guide to make RNA, also called mRNA. Today we're going to look at what does RNA do. RNA goes through a process called translation, and that's when it codes for those really important proteins. So here's another visual of these three processes. Replication is when DNA copies itself. What is replication important for? It's important for growth, asexual reproduction, and repair. Transcription and translation is important for making proteins. So today we're going to look at translation. We're going to watch a video first. And with this video, if you look in the top right corner, there is our symbol that some of these questions are going to be in your check for understanding in the Google form that you're on right now. So you can watch and answer the questions, or you can answer the questions all at the end.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
All right, so that was our animation for transcription and translation. So that was both of them combined. And we see at the end that translation creates a chain of amino acids. Now, if we said back here that translation is responsible for making proteins, why are amino acids being put together? That's because amino acids are the building blocks of proteins, if you remember that from our previous unit. So here's our definition of translation. You should write this down. Sorry, it's missing the little icon in the corner. Translation is the second step of protein synthesis and happens in the cytoplasm. A ribosome reads the mRNA and creates a chain of amino acids, which makes a protein. Pause the video here to write down the definition. If you think of the word translation, translate means to express in a different language. So in this process, mRNA is translated or turned into amino acids so that we can have our proteins. Now, in the video, it mentioned that the mRNA is read 3 bases at a time. 3 bases at a time is called a codon. So it's like smaller sections of DNA or RNA, and it's easier to separate these long strands of DNA, all these letters, into 3 bases called codons. So we're going to practice that right now. Here's our DNA strand. First, we want to divide it into codons. So if that's every 3, we're going to take a pen or pencil, or you're just looking at the screen, and we're dividing it every 3 bases. So that gives us 4 codons in our sequence. Then we want to transcribe it into mRNA. So this is really similar to the do now. So ATG would transcribe into UAC, GCT would transcribe into CGA, TAC would transcribe into AUG, and AG and T would transcribe into UCA. All right, now we can take our mRNA and we can turn them into amino acids. So this is done by using a chart called the Universal Genetic Code Chart. So we're going to show you how to read this and how to take mRNA, so this bottom strand here, and turn it into an amino acid. So if we have a DNA strand that reads GAA TGT, the mRNA that would be transcribed is CUU ACA. Now, to figure out what the amino acid is from these 2 codons, we look at our chart and we look at the first letter of our first codon, which is C. So looking at this chart, if you look at the left side, it says first base. Our first base is what? It's C. So that brings us to this box here. All right, so we're in this row. Now our second code or base is U. So if you look at the top row, we have UCAG. So we need to be in this column here. So if you cross these 2, that brings us to this box right here. Now, what's the last and final letter of our code? That would be another U. So we look into this box and we find CUU, which is right here. Knowing that our mRNA is CUU and it's in this box, over here is where we find the amino acid. Your amino acid is represented by 3 letters. It's the beginning of the word for the amino acid. So for our first mRNA codon, the amino acid that is produced is LEU, which actually stands for the amino acid leucine. Okay, let's try the next one. So our mRNA codon is ACA. The first base is A. So we go over here to the left side and we look for A. So I see U, C, A. Here we go. So that brings us to this row. Our second base is C. So I need to go to second base up here and find C, which is this column. So I know that my amino acid is going to be in this box. The third and final letter is A. So I look and I find ACA right here. Perfect. I know I'm on the right track. So our amino acid that's produced is THR. So I would write over here THR. And that actually stands for threonine. It's the abbreviation for threonine. So from a DNA sequence, we have created a chain of amino acids by using transcription, and mRNA to amino acid is translation. All right, now it's time to check your understanding. You're going to use the genetic code chart to find the amino acid for the following mRNA codons. So UCA, GGC, and AGA. You're going to go and fill this out in your Google form. It's 3 separate questions to check your understanding. You can pause the video now and answer it, or you can answer it at the end. So, reviewing the steps for translation, step 1 is mRNA moves into the cytoplasm and finds a ribosome. That ribosome is the organelle that's responsible for reading the mRNA. Step 2, your ribosome then takes the mRNA and reads them 3 bases at a time. Those are called codons. And then it starts to link the amino acids together. So here we have 3 codons. I'll circle them. 1, 2, 3. And then this is your chain of amino acids that's being linked together. This is happening in the ribosome, which is in the cytoplasm of the cell. Step 3 is the chain of amino acids fold together to make a protein. So your amino acids fold over each other, and then we have our final protein. So in your assignment for lesson 2, you will get a series of DNA sequences that you must transcribe into mRNA and then translate into an amino acid. This question specifically is in your Google form to check your understanding, and I'm just letting you know this is what your assignment will look like. So we'll do the first one together. So I have to turn DNA into mRNA. So T would pair with, you can do it with me if you want, A. A pairs with U. C pairs with G. So now that I have AUG, I have to use my genetic codon chart. And in your check for understanding, you'll have it with the question. And for AUG, the amino acid that's made, so I have A, no, that's in this row, U, which I know is in this column. That brings me down here, and I see AUG right there. The amino acid is MET or start. Now, when you see start, what do you think that means? Start would signify the amino acid that starts the beginning of the protein. You look at your chart, you'll also see one that says stop. You'll see 2 that says stop, actually. What do you think that means? The stop amino acids are the last amino acids in the sequence. So going back over here, my amino acid is MET. Right, so we're gonna have you guys fill out the second one in your Google form. And that's how we turn DNA into mRNA through transcription and mRNA into an amino acid through translation.